Good day, everyone. I remain Itudu Matthew Ocheleka. This is the continuation of the other lectures. What I want to explain here is how to make the writing of the structure and the labeling very easy for you as a student. Then I want you to look at this. I have established from the last class that purine, they have two rings joining together. This is a ring, this is another ring structure, another one, making two, and we call it bicyclic to have adenine and guanine. In purine, this is a good example, but in pyrimid it's not like that. Pyrimid we have cytosine, thymine, and then uracil. It is not like that. But now that you know the difference, now let's start with the cytosine. How can we label the cytosine? I've already told you that target the functional group. Target the functional group. How many functional group is attached to this cytosine? It's two functional group, the amide group and the carboxylic group. The amide group is carbon position number 4, while the carboxylic group is carbon position number 2. Now you come now and then you look at it now. The rest other ones now, I want to also give you a simple way on how you can write it out and label it clearly so that without you not bothering yourself or cramming it. Look at this hydrogen. Hydrogen should be attached to what? And nitrogen, this is nitrogen. Nitrogen should be attached to 3 hydrogen, just like in the case of ammonia. This is one hydrogen, second and what? Third, as in the case of ammonia. Look at this another nitrogen, 1, 2, 3. Then here we have carbon atom and we have carbon atom. And this double bond means there is shortage of 2 hydrogen. That is after you have attached these 2 functional group, amide and carboxylic group. Now thymine now. How many functional group do we have in thymine? We have 3 functional group. It's 2, but it's appearing in 3 places. Carbon number in position 4, we have carboxylic group. In position 2, we have carboxylic group. And in position 5, we have a methyl group attached. So having positioned the 3 functional group here, you know now that if here is double bond, there's no way you can have double bond here, just like the case of this one. So if here is double bond, as the carboxylic group is attached, then here will be a single bond. If here is a single bond, there's a nitrogen here, you already know that what? 1 nitrogen will be here, so that it can have 3 hydrogen attached to what? 1 hydrogen will be here, so that it will be making 3 hydrogen attached to the nitrogen. The same thing here and the same thing happen here. Then look at uracil. How many functional groups do we have in uracil? We have 2. It's 1, but it's appearing in 2 places, which is position 4, carboxylic group, position 2, carboxylic group. Now, since you have carboxylic group here, and if nitrogen is heavy, what are we supposed to? If nitrogen, we are supposed to have 3 hydrogens, and also 1, 2, and what? The third one, which is what? 3. So that is it. Then look at this one. What are we supposed to have here? We are supposed to have a nitrogen here. Then how many hydrogen attached? 3, which 1, 2, and 3. In case you look at it down here and you are thinking that it's another functional group as N and H. No. If the nitrogen is here, 1 hydrogen here, another hydrogen here, and look at another hydrogen very close here. That is how it appears. Now moving to adenine. Adenine, that bicyclic means 2 ring structure joined together, and we call them purine bases. Adenine represented with letter A, and we are saying, look at the functional group in adenine is only 1. Only 1 is the functional group in adenine. Only 1. And then if you have positioned it at position 6, the rest of that one now you already know where to put your nitrogen, because you know the number of nitrogen that is in purine. You already know from the last class, and you know the number of the carbon atom. So now that you know the number of the nitrogen and the number of the carbon atom, follow the numbering. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7, 8, and 9. Then, if you are able to put it so, then attach your amide group here as this functional group. Then you are good to go. How many functional groups do we have here? We have in guanine we have 2, which is the carboxylic group and then the amide group. Then you know the number of the hydrogen and you know the number of the carbon atom. Place it the way it's supposed to be, and then you have what? Your guanine. I believe this class is helping somebody. We are going. Let's know the source. How are we able to have this? How are we able to have it? I want to explain something to you very striking to you. In 1948, John Buchanan traced the source of all the 9 atoms of purine ring. The 9 atoms of purine rings, that 1, 2, 3, 4 to 9, is able to trace the source of it. And this is what John Buchanan said. He said at nitrogen 1, the source of nitrogen 1 is from the aspartate, which of amino acid. There's amino acid we call aspartate, that is the source. Then nitrogen 3 and nitrogen 9 is from glutamate. It's also an amino acid. Then from carbon 4, carbon 5 and nitrogen 7 is from what? Glycine. And then from carbon 6 is from CO2. Then carbon 2 and carbon 8 are from tetrahydrofolate. Don't forget, it's from what? Tetrahydrofolate. Tetrahydrofolate is the meaning of THF. That is the source of what? Carbon 2, carbon 8. So this will help you. This is how we are able to have the 9-member ring of purine. In case you are asking yourself, where is it coming from? This is from the food you eat. And you know, so we have essential amino acid and we have non-essential amino acid. There's amino acid generated by the body. There's the one from that is from the food we eat. The one from the food we eat is there, and we call that one essential amino acid. The one that is generated by the body is non-essential amino acid. So as all the amino acids come to the body, it's able to donate and contribute to the 9-member ring of purine. That is what we are talking about, and we are proceeding right way. Now, the difference between DNA and RNA structure. Look at the difference. This is what? RNA, and we call it what? Ribonucleic acid. You see, it is only one single-stranded RNA. It's only one single-stranded. And then look at how they are giving an example of it. Look at it. And then look at this one. It is what? Double-stranded. And all that we are seeing here, all these things that we are seeing here, as you are seeing the label, they are what? Nitrogenous bases. All this label you are seeing inside, all this label that you are seeing inside, they are what? Nitrogenous base. Why this one that is holding it, wrapping it around, they are the sugar phosphate group. Now, I earlier told you before that I'm going to tell you the difference between DNA and RNA. Now, this is the functions. One of the number one function that show the difference between deoxyribonucleic acid and the ribonucleic acid. One of the function is what? Why deoxyribonucleic acid is for storage medium of genetic information. Why the ribonucleic acid is for transmission molecules. Transmission, don't be subscribed, because it's DNA that is transcribed to messenger RNA. That's why they call it what? Transmission molecule. Transmission means it is the information in the DNA that is transmitted to the messenger RNA. So it's for transmission molecule. And it may be storage medium in primitive organism. Then structural features. One of the difference we can see experiences in what? In their structural features. We say the DNA have what? Beta form double helix consisting of what? Long chain of nucleotides. The nucleotide, long chain of what? Nucleotides. why this RNA is what? A formed single-stranded helix consists of what? A shorter chain of what? Nucleotide. Means this one have the array of nucleotide sequence that is in this is longer than the one that is in DNA is longer than the one that is in RNA. Now the base sugar composition is another differences, and then we said that was the ribosugar phosphate backbone, and that is what we mean that the nitrogenous base that we have in DNA is different from the nitrogenous base that you are going to have in what? In RNA. This A is adenine, G is guanine, C is cytosine, T is thymine. These are the nitrogenous base we have what? In DNA. I told you earlier. And these are also the what? Nitrogenous base we have what? In RNA, which is guanine, cytosine, and uracil. So if you look at it, the differences here is what? Thymine and uracil is the differences here. Why in DNA you have thymine and you don't have uracil, in RNA you have uracil and you don't have thymine. That's what we mean. Another one is in propagation. DNA is self-replicating. Self-replication means, DNA is able to unwind itself for you to be able to have what? A single-stranded replicating DNA for the protein to be able to bind to have a photocopy DNA. Like in the case of meiosis, for you to have a new baby born resembling the parents at the cause of short division of meiosis. These are self-replication processes. But if we are talking about protein to be formed, a child growing, developing muscle, growing bigger, that is the division of the mitosis. We say was synthesized from DNA. That one means DNA have to be transcribed to RNA, and so forth. That's what we mean here. Then in reactivity, I told you about this before, that in reactivity we said CH bond make it fairly stable. So when it is bounded to hydrogen, it is what? Fairly stable. Okay? But when OH is bounded, it is what? Reactive. Means OH is bounded, it make it more reactive than what? When hydrogen is what? More is bounded. So I know this class has helped you. Now we are moving to functions of DNA. Functions of DNA. One of the number one functions of DNA is store genetic information. The genetic information of the DNA, it is stored in the nucleus, and then in the nucleus it is transcribed to messenger RNA, and it finds its way out of the nucleus to the cytosine, in which it is transcribed to protein. So it is the genetic information that is in the nucleus that is able to allow the synthesis of protein. And it is still the same that genetic information that is in the nucleus in form of a DNA, DNA that is also to carry out DNA replication for you to have a new daughter cell, for you to have a new husband looking like the father and the parents. So that is what we mean by store genetic information. 2, maintain growth and repair. This can happen at the point of what? Mitosis. That a smaller person grow to become a big muscular human being. At the cause of what? The cell keep dividing, so that the human being can be able to grow. So it maintains growth and repair. And as a cause of cell dividing, old cells will divide to form new cell, and so that's why it keep dividing, and it keep dividing. Number 3 is control all cellular activities. All cellular activity as in the body, since all the information is in the DNA control every cellular activity in the body. So that's why that the body is wired in such a way that information at one point communicate to another information at another point. There is nothing that is out of control in the body, because all the information is wired in the DNA. And so all the cellular activities, they are under control. Number 4, it contains protein codes. There's what we call